Okay, good morning and welcome to what I think is the first Complex Human Data Hub seminar for this year. University of Melbourne acknowledges the Wurundjeri people as the traditional owners of the land on which this meeting takes place and their elders past, present and emerging. It's my particular great pleasure to welcome Roger Ratcliff back to the shores of Australia after a gap of 3 years. Many of us will remember that the last time we saw Roger was at the Coogee Beach Mathematical Psychology Conference in 2020. That was just when COVID was an impending looming threat on the horizon in February 2020. In March 2020, the New South Wales Health Department unloaded a cruise ship full of positive COVID cases and unleashed a pandemic on the country. And you all know very vividly what the story has been since. And so it wasn't clear when, if ever, it would be possible to have meetings like this again. So it's kind of great to have Roger back with us again. Now, anyone with a pulse will know the extent of Roger's contribution to mathematical psychology, which I think over decades has been unmatched in the discipline, and not merely within psychology, but the impact outside psychology in disciplines like neuroscience and economics. So I won't rehearse all of that. One of the things that a lot of you won't be aware is that Roger's been an incredible friend to mathematical psychology in Australia for several decades. So in 1998, we did a special edition of the Australian Journal of Psychology on mathematical psychology in Australia, which Steve Lewandowski edited and Roger wrote a foreword for. And it was just a way of mathematical psychology in Australia raising the flag. We're here, we do this stuff and we're actually really good. We are of international quality. And I think since then, Roger has been, I think, a tireless ambassador, possibly inadvertently, but nevertheless, for mathematical psychology and for the Australian Mathematical Psychology meeting, AusMathPsych, which has become, in its own little way, quite an international conference. So we have had over the years many international visitors from North America and Europe. And I think a lot of that has been word of mouth that was originally seeded by Roger. Roger saying to people, look, this is a really great meeting. They're high quality, there are nice people, you should come. And indeed, we built it and they came. So with all of that, it's a great pleasure to welcome Roger back. So,
yeah. It's mainly for the cricket game and soccer game at the AusMathPsych. That's the most important thing. So there is a soccer game on like a Thursday or something. Yeah, so I hope you'll all be out there and get humiliated by the non-Australians. That's the whole aim. So why did you make this go away, Sasha? You can't see the title. Oh, no, leave it. It's okay. I don't think I've got very much up there. See if you can. Challenge. I've heard that before. So one of the things in the US, is this too loud? Okay. One of the things is that most of the funding for research, most of the big funding comes from NIH, National Institutes of Health. If you look at an NIH grant, it's usually 3 or 4 times larger than a National Science Foundation grant or an Air Force grant or wherever. And so I've been funding from there. And I've always had this sort of interest in applications to take this modeling into clinical domains. It's only one of many things I've tried to do. So at one point I got roped into an NCI, National Cancer Institute meeting on cognitive deficits in cancer treatment. So what I'll do is mention a little bit about that, talk about processing speed. This is all about neuropsych testing. Then issues in using reaction time modeling in this domain, and saying this is an area that's really ripe for us to look at as a field, and just to discuss how little impact we've had on this. I'll present some data from a paper that where we used modeling to discriminate memory disorders, that's mild cognitive impairment, Alzheimer's, from controls. then a review paper by a graduate student that I'm on ADHD, and then something that didn't work very well, number line task, trying to do the same thing as in 4. Then I'll mention some problems with online data. I know this is becoming very popular, and a lot of online data is really, really bad. So you've got to look at it, and there's a very simple way of looking at it, and then maybe discuss things at the end. Let's see if this, yeah. So everybody in the research community knows, or used to know, that NIMH pulled out of behavioral modeling research. They were a major source of funding. And then we had to convince other agencies. NIA, National Institute on Aging, was always happy with that. But these other institutes want you to have a patient population that is represented there. Luckily, I'd been doing this stuff with aging since about 2000. So I didn't need to come in as an outsider to the field, which was good. But then what we have to do is try and convince them that the work is useful or important. So if it's really important, we should see impact in other areas, practical impacts. So Thomas Insel is the guy that pulled NIMH out of behavioral research. And so there was a book written in 2022, I guess early in last year, about a year ago. He helped allocate 20 billion in federal funds and shifted the focus of NIMH away from behavioral research and towards neuroscience and genetics. And so he says, we should have been able to help things, but it didn't. And now he regrets the size of the shift. Yeah, after getting rid of people like Schifrin and Nosovsky had trouble getting grants after that because they were funded through NIMH. So the major problem is neuroscience and genetics are really difficult. It sort of turns out that they involve interacting systems with feedback where, you know, just very, very complicated. Genetics is like that too now, apparently. My NIA program officer says things like it was a big scam, all this genetics. They put billions and billions and billions into it, and almost nothing has come out of it of use. The promise wasn't there. So modern, post-1970, I say modern since 1970, has had almost no impact on the domain of neuropsych testing and diagnosis. So if you have, how many of you have come across neuropsych testing, have looked at it in detail? Yeah, you'll see that cognitive tests used to assess functions of working memory, episodic memory, executive functioning, speed of processing, have nothing to do with the concepts that we use in cognitive research. There's a book in 2008, Information Processing Speed in Clinical Populations. I think they mentioned Sternberg task and model. That's the latest modeling. There's Intelligence and Speed of Processing, a review of 50 years of research. No mention of anything modern. So last year, I zoomed into a talk from a neurologist at Mass General. This was something that somebody from the National Cancer Institute was running on cognitive deficits. And he presented research that used exactly the same kinds of tests from 40 years ago. Nothing new, no penetration of what we do. But then you get National Institute on Aging has this cognitive impairment in decision-making. So they put out a call. It was a one-shot deal to get a center together to look at deficits in decision-making capacity used for screening and those kinds of things. So there is some hope. So the lack of impact, one issue is big money in the infrastructure of testing. So out of this, you can get Medicare price data, maybe 800 million a year is spent on neuropsych testing. Money spent on grants, all this private stuff. You can sign up for neuropsych tests and it costs large amounts of money. I didn't do a comprehensive search, but I just noticed large amounts of money. So there's a strong interest invested in keeping the status quo. Gail McCune and I have tried 2 or 3 times to examine deficits under chemotherapy. And we have, so here are some of the issues. So this was to talking to some people who are doing deficits in chemotherapy in a Zoom meeting. We need data from individuals before, during, and after treatment. So before, it's usually people get the diagnosis and then within a week or 2, they're on chemotherapy. So if somebody found out they needed chemotherapy, they don't want to be bothered with the experiment. So one option is make testing part of the treatment plan, but that requires a lot of work and it needs doctors to be heavily involved in this. During treatment, maybe a third to a half, no, I think it's about 20 to 30% have something that's like long COVID out of chemotherapy. It's called chemobrain. They don't want to be tested if they feel bad. So you get attrition of the patients with deficits. So this becomes a problem. You need engaged physicians and not just research assistants that go along, which we had, sees the doctor and begs them to be in the study. And we need to figure out what tasks, what tap into what deficits, and then run enough trials for modeling. And so often you might get 10 minutes of testing on the end of something else, and that's just not going to work if you're going to do modeling. That amount of data is not enough. So this is how the neuropsych world sees cognitive processing and testing. So this is from Nature's Review, Cancer, 2007. Memory is working memory, so the ability to temporarily store and manipulate information, and they've even located the brain areas where this happens, which of course explains everything. Yeah, in America, that's called irony, and in America, they don't get it, so you've got to be a little bit careful. Episodic memory, the ability to learn and retain new context-dependent information. Remote memory, then you get modality specific. Executive function, planning, rule acquisition. Yeah, where's the model for this? I mean, there's no good model for that kind of stuff. It's processing speed, which should be reaction time, speed and efficiency with which information is used in completing a task. And they've located in the frontal subcortical. That's where reaction time is. Yeah, it's weird, but this is the state of the art, and this is one of the big names in that field of cognitive deficits in chemotherapy. But you get the sense. Reaction time is the time it takes for a person to react to a stimulus, pressing a button when a light goes on. Period. Yeah, you get this. The challenge is to go into one of these domains and ask what the processes and representations are in the tasks. That's what we're trying to do. And we want to do this in tasks that represent memory and perception and how they relate to deficits. So one of the questions, this was a program officer at this meeting in 2011. She wanted us to get specific. So are neuropsych tests sensitive enough to detect mild cognitive impairment? Sort of ecological validity. Do these things assess a person's behavior in real world settings? So she was asking things like, well, if we get lower neuropsych test scores in a control group, we get 2 standard deviations below the norms, or 3 subtests below the control group mean, or 2 subtests less than 1.5, or 1 less than, -1 standard deviation on 2 measures. So you can see how she was thinking this. The problem is it's still using these same neuropsych tests. So what is speed of processing? Well, speed of processing involves digit symbol coding. That's where you get a row of letters and symbols, and you present with a symbol and you have to write the letter, usually pen and paper these. Trail making tests, you get A, 1, B, 2, and you have to draw lines between them. Color word interference test is kind of a Stroop test. Groove pegboard, I keep forgetting what that is, but it's placing pegs or something. So this is processing speed. This is not the kind of reaction time stuff we study at all. Reaction time is the continuous performance task. It's a go, no-go task where it's sort of like a vigilance task. You have, say, 2 stimuli and you hit a button if one occurs, but not if the other. And the probability of hitting a button is like 25%. So, oops. So if you're doing assessment, there's a list, say, of speed of processing tests, and a practitioner simply selects 3 or 4 from the list. You get scores according to the rules that are calculated. There are formulas for doing this. It might be the number correct in some fixed time. That's a standard measure. Or stimulus duration for 75% correct. Then you combine these. Then you compare them with control groups with z-scores. If you're very sophisticated, you might throw them into a factor analysis or structural equation modeling. And there we understand how cognition is changing. But scoring a simple test or a brain network activity can't equal, say, working memory or executive function. We just, there's this massive disconnect there. So what, oh, and then this is an example of one of the major problems. The big problem is to do with the size of deficits relative to individual differences. So if you have, say, people functioning in the bottom 33% functioning, these might be the ones with deficits, there's always some overlap. The group might go down if you go from baseline to chemotherapy to recovery. So the group goes down, means go down like this. But where's this individual? Is it part of this group or that group? You know, you can't really determine. It's okay for group kind of data because, you know, once you get standard errors instead of standard deviations, you can see the group moving up and down. So you can study group behavior. But the key is to do individual diagnosis. You need a test that's sensitive and produces a large performance deficit even under mild impairment. Okay. So one thing I want to be clear about, the existing construct, data does not equal function. That's what all these neuropsych tests are. And, you know, you read in the paper, you know, exercise may help aging. You know, caffeine may help this. There's always this may help. Nobody says exactly it does help. And it's all about, you know, data and function with no model or theory of what's going on in between. So do modern cognitive tasks help? Do models help? There's such a lot of work going on. I sent Philip, there's a review by Tolving and Madigan, 1970. How many people have read that? You really need to read the first 20 pages of that. It says essentially that they reviewed about 350 articles and they say, you know, like 2/3 are completely worthless, you know, maybe 10% are worthwhile. That was back when there weren't very many papers. I'd say now maybe 5% of the papers that are published are worthwhile. Then this gets me to, who cares about the replicability? Say there's 95%. Why are we spending all this time on it? That's another argument, but sort of drives me nuts. Is speed of processing a function? Brain and genetic expression, as I said earlier, are interacting systems with complicated network feedback loops at different timescales. And I don't think we have the tools to understand these systems as yet. So can we do anything with the kind of modeling we do? So this is standard diffusion modeling stuff. What I'm going to do is do the two-choice model, noisy accumulation of information. It explains all the variables, correct and error reaction times, and their distributions and accuracy. I'll describe work in memory, and then we'll go into memory disorders. So we go from college students to 75 to 90-year-olds and how memory changes with age, and then this work on memory disorders that just came out a couple of years ago. So another problem with psychology is that a lot of work, especially empirical work, you either look at accuracy or time and don't look at both. And yeah, right at the end, I can show you some data that I'll talk about at the meeting where accuracy and reaction time give completely different patterns of results. And when you put them together, you find something different again. Diffusion model addresses time and accuracy. It's modeled over a wide range of tasks. It's been now applied to all these different domains, and a lot of them are not very good applications. So when you look at these things, you've really got to go in and use our standards, you know, numbers of observations, standard errors, whether the model is fit correctly, et cetera, et cetera. Yeah. So one thing in modeling is we have a criterion. The model has to fit the data. If it doesn't, then you can't use the model. In neuropsych testing, you just apply the test. There's no criterion if they're appropriate or anything. So this is one of the important things about cognitive models. You want to make sure they work before you can apply them. There's no equivalent test. So usually I provide group fits to accuracy and reaction time distributions, and then I've started doing individual fits of every individual and condition to accuracy and reaction time and plot them against each other, and this shows any systematic misses. So I've been getting more and more interested in presenting data in a way that exposes everything. There's something wrong. You know, most of the graduate students sort of edge things and hide things. I was just working on a paper by a postdoc, and I think he hid something in there, something that didn't work quite right. So you've got to think about it as if any of you are graduate students, that don't try to please your advisor, but just lay out exactly what's going on. you've got to get consistent in estimates of individual differences. So if you look at reinforcement learning and Iowa gambling tests, as far as I know, there's no general agreement that the model parameters that come out of those measure individual differences in a reliable kind of way. I might be wrong on that, but I think there are conflicting models in some of these tests, and they don't really converge on the same kind of thing. Reaction time, when people have looked at other models, you seem to get about the same results out, the same kind of effects. And when dealing with, say, categorization models, word recognition models, memory models, you ask the question, have these models been fit to individuals, and have people tried to understand individual differences in the models? And most of the time, not. I don't know of any of, say, the SAM model or REM model have been applied to individual differences. And you try to interpret parameters in terms of abilities or things like that. It's a real challenge. And, luckily, I wouldn't be saying this otherwise, this model does. So examine the effect of aging. So we have a process that accumulates noisy information over time towards a correct boundary, an error boundary. This would be, if the drift rate is up here, it probably means this is the boundary for a correct response. Say in memory, it would be an item that was studied before, an old response, and down here would be a new response. And you've got a drift rate up here. Sometimes you hit quickly, sometimes you hit slowly, and that produces the reaction time distribution shape. And sometimes because of noise, you hit the wrong boundary by mistake and it gives errors. We have boundary separation as one parameter. If you're manipulating speed accuracy trade-off, this is the thing that changes most to accommodate speed and accuracy effects. We've got drift rate, which is evidence from the stimulus. And then there's non-decision time. That's the time to encode the stimulus, access memory, or extract the features of the stimulus that are relevant to the decision. So if I present a string of letters, it could be, you could make a decision on whether it was red or green, whether it was upper or lowercase, whether it was a word or a non-word, whether you'd seen it before. So when you encode a stimulus, all these features are there. And what you've got to do is sort of extract the ones that are relevant to the decision and do that. And that's something we don't really have much of a handle on, but it's part of non-decision time. So you've got encoding, that process, and response output. And so, So we focus on these parameters for individual differences. They're estimated the most accurately. Boundary separation, non-decision time, drift rate. We fit the model to these across conditions, and it produces values for these components of processing. So we have a criterion, non-decision time, and evidence. So I'll talk about two experiments in aging and then go into the memory disorders. So recognition memory, you have words that are high and low frequency in English. You study a list of words and then get a test list. And you respond old or new to each item in the test list, each test word. So a test word comes on, did you see it before or not? Very simple. Lexical decision, you get a string of word and you string of letters, and you respond word or non-word to the string. And the manipulation you have here is high, low, or very low frequency in English. Non-words in the experiment we've run are English-like non-words. So you take words and change a vowel or something like that and turn it into a non-word, and then make sure you didn't turn it into another word. So aging, the data fits them, the model fits the data well. Older adults have wider boundary separation than young adults. So with age, and this is going from college-age students up to 60 to 75 and 75 to 90. We usually have 3 age groups. Older adults have wider boundary separation, longer non-decision time. But in these tests, they don't have lower values of drift rate for recognition, memory, or lexical decision. And we get this really nice separation. We get no effect on drift rate, but an effect on these two parameters. But with IQ, we get an effect on drift rate, but not an effect on non-decision time and boundary separation. And then if we use a different task, and this is another paper, associative recognition, where you study pairs of words, and then you have to say, were these two words in the same pair or not? Performance essentially drops to floor by the time you get to 75. So if you've got to remember whether things go together or not, The only way to do that is to write them down and remember where that was put, that piece of paper. So you keep a note of where you keep the notes of the piece of paper. Infinite regress, yeah. Individual differences. Accuracy and mean reaction times correlate from task to task, from recognition to lexical decision. Drift rates, boundary separation, non-decision time correlate with each other from task to task. So if you get boundary separation for a group and one task and boundary separation for the same group and another task, those two correlate, and it might be like 0.5 or something like that. In memory, we get cued recall, free recall, drift rates for item recognition and associative recognition correlate. So if memory is good on one of these tasks, it's good on the others. This starts to break down when you get to 75 to 90. The correlations aren't as strong. And drift rates correlate between lexical decision and recognition for all age groups. So what this is showing is that there's a nice story for aging and IQ. There's separation of the effects and parameters. And it identifies strong individual differences that can be used for maybe applications like the next one. So we ran this experiment, and it started about 15 years ago with item recognition lexical decision on memory disordered patients. These are ones in the memory disorders clinic with people with Alzheimer's and mild cognitive impairment, as diagnosed by clinicians. Now, the clinic had one doctor when we started out, and it was across the road, and we get like one patient every couple of weeks, and that went on, and then there were funding went down, and we didn't have time to do this, so there was a lapse, and then when we got some funding from NIA, we cranked it up again using exactly the same list. And so the idea is to see if a diffusion model can fit the data, and then what changes between memory disordered patients and older adult controls. And then see if diffusion model parameters can be used to discriminate between the patients and controls. So they were tested on about 15 minutes of lexical decision and 25 to 30 minutes of recognition memory. Enough to get, maybe 400, 500 observations, somewhere, a reasonable amount for modeling. So they're diagnosed in the clinic by neurologists. We got 105 memory disordered patients and 57 older adult controls. These were tested exactly the same way, just like in a clinical trial, except we didn't have to go through paperwork. So we had research assistants testing them, sitting in there, watching what they were doing. And I think this is important, as I'll say a bit later. So in recognition, we get from the older adult controls in the same age group, go up from 0.18 to 0.21. There's a significant change in boundary separation. There's a decent sized change in non-decision time. And there's a drop in memory performance and drift rate, which represents evidence in memory. And because they're memory disordered, and this is a memory task, good, it sort of looks right. Lexical decision, we get the same kind of difference in boundaries. Same kind of difference in non-decision time, perhaps attenuated, and only a smaller drop in drift rate evidence being used in making the decision. For those of you that know this, the fits are okay, they're not great. These are the quantiles of the reaction time distribution plotted against accuracy or proportion of choices. So this might be high frequency new words, low frequency new words, and that's the 0.13579 quantiles. And you can read reaction time distributions off this by drawing equal area rectangles, which represent equal probability mass between the quantiles. Turn your head sideways and you've got a distribution. So you can sort of get a good understanding of how the distribution is changing, whether it's shifting up or whether it's spreading or what. So that worked. Then what we did was try linear discriminant analysis. So what we do is we throw in model parameters and see if we can get a weighting on those that separates two groups of subjects. We also did it for logistic regression and support vector machines. So one thing to note is the clinical accuracy of classification is 85 to 90%. So if you open up the brain of somebody who's been diagnosed with Alzheimer's, it's not that accurate. It's 90% is about the limit. They get people that they think have Alzheimer's and they don't, and vice versa. So linear discriminant analysis finds a weighting on the parameters. And what we do is you can fit all of them and look for discrimination. But what we do is a cross-validation. We might run 100 classifications where we randomly select 2/3 of the data and test on the other third. So this would see what would happen if you train on one group and then you test on another group to look at accuracy. And what we find is we get about 83% correct accuracy based on the recognition task, 76% based on lexical decision. And it still remains about 83% if you train on both tests. So lexical decision didn't really add anything to this. And in the discrimination, it was a bit of a surprise, but boundary separation is weighted most, drift rate next, and non-decision time next, even though there were large differences between the groups and this parameter in particular. So they were tested in the same way. And then we added 88 subjects that weren't run in exactly the same way to see if this helped discrimination. They didn't have lexical decision. They were tested on recognition. We still get about 83% accuracy. So we tried a lot of things on this. So here's a classification. I think in the article, there are about 20 lines trying all different kinds of things. So these are cross-validation, the ones you really want to look at. Full discrimination says if you throw them all in together, you get this. But full discrimination will overfit the model a little bit. And so if you try somebody new, performance will go down. So cross-validation is the way to do it. So we have memory disorders versus memory disorder controls and this other set of 88 subjects, and we get up to 83% correct. We look at Alzheimer's versus mild cognitive impairment, 70% correct. Alzheimer's versus the controls, yeah. But if you look at Alzheimer's, very quickly you realize there's something wrong. So it's not like a major feat of getting 91% correct. Mild cognitive impairment isn't that good, 75% correct, but in the controls, there might be people with mild cognitive impairment, and in the mild cognitive impairment, some of them may not be correctly diagnosed. Then we were asked to run what happens if you collect less data. So we collected less data and did memory disorders versus controls. It would be something like this, and performance drops to 77%, 78%. So with limited data, you can still do okay. And then we were asked, well, why don't you do it with a modern, sophisticated method like Bayesian diffusion model package, HDDM, hierarchical, do the hierarchical modeling, and performance was a little worse. There's a problem with HDDM. You've got to classify the people in two groups and train. It takes about a week for the model to produce a hierarchical analysis. And then what do you do with a new patient? How do you assess which group they go in? You throw them in and train the whole system again. I don't know, it sort of seemed a little silly after the fact, but reviewers wanted it, so we did it. In other words, things did not get radically better once we used the hierarchical Bayesian method. So these are dots representing individual subjects. So here are the Alzheimer's, the mild cognitive impairment. So this is what the classifier says. These are classified as memory disorders. These are classified as controls. You can see there are 3 Alzheimer's that were classified as controls. 52 out of 56, 76 diagnosed with mild cognitive impairment were called mild cognitive impairment. And just a few, like 7 out of 57, were called memory disorders when they weren't. And some of these could have been genuinely ones because all we had was the mini mental test, and they had to be above a certain criterion, but it doesn't guarantee they didn't have memory problems. We can also do a 4-way classification. It does about as good, linear discriminant analysis. I think we'll do that. And it was calling most things mild cognitive impairment. Very few reached the level of Alzheimer's. And none of the controls were called Alzheimer's in that group. So that was trained on 4 categories. So here are the actual scores out of the linear discriminant analysis for the Alzheimer's. And this just becomes signal detection theory. You can put a criterion across here and look at the hit rate and the correct rejection rate. And you can move the criterion around until you get the maximum that you want for your comparison, whether you want to be conservative or liberal on calling somebody memory disordered. You probably want to be up here somewhere so that if you're a control, you're not called memory disordered by mistake and put people through pain. But you can see the scores, there's a range of scores, and these are the three. I think these three here were the ones that were of the Alzheimer's group that were called controls. They're not way down here like most of the controls, they're still up here. And then, so you can do histograms of the patients on the regressor. Controls look like that, which is just like signal detection theory. And you can get hit rates. And it turns out I didn't know this till about 7 or 8 years ago when I visited, where is it? Washington University. Talked to some Alzheimer's people. The hit rate is called sensitivity and the correct rejection rate is called specificity. So if you're looking at neuropsych testing, these words come up. And it took me a while to figure it out. What the hell are they talking about? You know. But they've got their own words. And the thing is, they don't realize that these criteria can be moved around. So they seem to be fixed numbers in that world. All right, so now example 2. We wrote Nadia did this incredible review of ADHD model-based approaches. It was really kind of exhausting, especially working over the paper. It probably took more effort on her part to write it, but I had trouble making comments. So there are various test domains, and these are the ones that are most common. And this was classified the model-based studies in ADHD. The cognitive flexibility, selective attention, working memory, time perception, sustained attention, inhibitory control. These are all supposed to be things that can go wrong in ADHD. And selective attention, ability to maintain focus on a stimulus in the presence of distractors. Ability to maintain attention, withholding ongoing actions in the presence of new information. And the tasks used are task switch paradigms, Stroop tasks, Nivon tasks, flanker tasks, anti-cascade tasks for cognitive flexibility, and go down to inhibitory control, stop signal tasks, Stroop tasks, continuous performance tasks, and so on. And these were the number of studies that use these tasks in applied models, either reaction time distributions or diffusion models. So anybody see what the big problem is with this? There's a really big problem. Yeah. Nivon task was in 3 constructs. The flanker, Stroop, and continuous performance tasks, these are no-go, no-go tasks, were in 2 constructs. So there's no one-to-one correspondence between tasks and constructs. This makes the whole thing kind of a mockery, if you ask me. So Horowitz, who's worked with Jeremy Wolf and is a program officer at National Cancer Institute, published a paper, Understanding the Profile of Cancer-Related Cognitive Impairments, a critique of meta-analyses. So one of the cheap ways of doing research in these fields is to do a meta-analysis. All you do is look up effect sizes in papers and do it, do a search and then write like crazy. So there are 7 meta-analysis studies of cognitive impairments in chemo. So every domain was impaired in at least one meta-analysis and unimpaired in at least one other. And in the study, single tasks were assigned to multiple domains. So this just showed that in the chemo domain, again, there's no correspondence between tasks and function. And we published this paper. We sort of got annoyed enough to publish a paper in Trends in Cognitive Science that just came out that goes through these things and lays out this argument and says, can we do better in modeling? So if you look in detail at these applications of models, problems arise because the tasks differ. Even the different variants of a task are run differently, but subject groups differ. The degree to which data are available differs. Even when multiple studies use the same task, details in the procedures differ to such a degree that they couldn't be compared unambiguously. So contradictory results can occur because of difference between groups, task specifics, differences between tasks, and so on. So if you want to get into the weeds on this, that paper is a good place to look for all the details of these things. She did a really good job of laying out what happens. So the question is, do commonly used tasks validly assess their target cognitive concepts? So terms such as inhibition, selective attention, inhibitory control are useful for talking about the possible locus of deficits, but the tasks used to assess these, like Stroop task, or sometimes they use the flanker task, or they use a go, no-go task. Are these useful for looking at what happens when you have inhibitory control? Like one example might be, I'm still wearing my pants, so I have inhibitory control to put them on. The day-to-day activities in inhibitory control are different probably from the behavior of these tasks. And one task is used to measure several concepts, so it's difficult to relate tasks to concepts. ADHD specific longer mean reaction times sometimes came from deficits in processing. That's informational perceptual processing, but sometimes they came from more cautious response strategy. In other words, in the diffusion model, it would be boundaries. So you've got mean reaction time differences, and some studies found it in evidence, and some people found it in boundaries. So that domain is kind of a mess, and the effects are all very small. If we get time, I could come back to this, but this is some of the more details. And then something that didn't really work that well, we were looking at numeracy and memory disorders. And we're going to use a different model, a spatially continuous diffusion model of the number line task. So the task is, you put your finger here on a touch screen, 2-digit number comes up, and then you have to move your finger to where it is on the screen. So it would be here somewhere. And so you look at the time from when you lift your finger to move, and you encourage subjects to move ballistically without, changing direction. And you look at the distribution of responses on the line. Okay, so here's our spatially continuous diffusion model. It's like a population code model. We assume a distributed stimulus representation. Evidence is accumulated to a threshold. So what we have in the model is what's called Gaussian process noise. So you take random Gaussian values, random normal values, and then you smooth them over the spatial scale. Over this scale would be number, like 20 to 100. And there's a parameter that represents the scale. So this has the property that any vertical cut, the distribution of the values is normal, but there's a correlation between the values that dies away the further between the two values. And so what we're doing is we're accumulating the stimulus information, the blue line, with different samples of noise on different time steps until one of the accumulated evidence point, somewhere on the line, accumulated evidence hits the decision boundary. Then we make a response output. So this is an alternative model on the spatial, and as I said, it links more to population code models that are very similar in the neural domain. So here are responses. We have 62 memory disordered patients and 51 control, which look the same. You can't really tell the difference. So here's what happens. So here numbers with them maybe 55 to 65. They tend to respond around, with a peak around 60. It drops off on both sides. So the results are what you'd expect. They're by and large, they tend to hit around the correct number. And there's sort of this W-shaped function where you might have an anchor point in the middle of the scale. So you get a narrow distribution. And we fit the model to data which have different standard deviations for each of the distributions. And we can get the same kind of pattern, and they match up pretty well. And then what we have is in the data, quantiles of the reaction time distribution, the 0.1, 0.5, the median, and the 0.9, the tail of the distribution. And we look at the model, and of course, the model produces smoother results, but by and large, the match is quite reasonable. Not as good as you'd want. If you look at these points here, these are up around 1400, these are around 1350, but hey, there's no competitor, so we're okay. And that was meant to be. All right. So here are results of the parameters. So this is exposing the whole parameter space. We have different standard deviations. These are with a mean of 5 and 15, ranges around these, up to 95. So the first thing is non-decision time is longer for the memory disordered patients. This is the height of those distributions. It multiplies the height of, you have a distribution with a standard deviation, and this multiplies the height of all of them to produce the evidence driving the process. And memory disorders are a little less than controls, but not much. And then this standard deviation is the mean of the standard deviations of all those distributions estimated from the model. So it's the mean of this row here. So that's 8.5. So there's not much difference in the spread. It's not like the patients go off and have wider spread. We also checked that the hitting the point. So we had initial trials where a dot came up and they had to hit it. And the standard deviation is about 8 on the scale, and the standard deviation for hitting the dot accurately is about 1. So it's not that these patients have, fingers that don't work too well. So we get that, and then we put all these in, and there are many more lines in the table, including the mini mental. And with cross-validation, we only get about 68%. So that's nowhere near the 83% that we got for the other task. And if we take out mini mental, which correlates a bit with drift rate and memory, we're about 65% correct. And it turns out in this case, logistic regression actually did a little better than linear discriminant analysis. And I'm not sure why. But this suggests the task is not a good candidate to discriminate patients from controls. And in the literature, there was this hint that maybe Alzheimer's patients start to get dyscalculia and their ability to do numerical things decays, but we didn't see it in this task. This task has been useful as well in development. If you look at development in Numberline, you get pages and pages of examples for teaching, and there are lots in the numerical cognition literature. This task is used about half the time in empirical studies. I just want to talk a little bit about online testing because we had a paper with Drew Hendrickson, used to be at Adelaide. So we want to do lexical decision and item recognition, the same tasks that we ran with our aging studies. And then we got 154 subjects, and then we had 199 subjects with 2 numerosity discrimination tasks. And in the memory and item recognition, we had an IQ test, which was culture fair test. And the other tests, we ran some subjects on IQ and some on math computation. And in the talk by the neurologist, he discussed the need to go to online testing because there's too much testing for individualized one-on-one. There's just, it takes 3 hours to do these neuropsych tests, and you have somebody who's been trained in testing talking to the person. This is why it costs so much. So, lexical decision, item recognition, about 90% of the subjects were good and 10% were erratic. We got aging effects replicating with boundary separation, non-decision time, and drift rates were correlated with IQ. Model parameters correlated across tasks. Numerosity tests, there were similar results, but about 45% of the subjects were erratic. Now, by erratic, this is what I did. All I did was plot every reaction time horizontally across the experiment. The vertical lines represent blocks of trials. The dotted line is about 300 milliseconds. So look at this subject here, making responses around 600 milliseconds, and then suddenly dive to about 150 milliseconds. It takes about 150 milliseconds for light to go from the eyes to the frontal cortex and then to the motor cortex. So there's no thinking in here at all. They're just seeing something and hitting a button, and they're at chance. So with these mechanical Turk subjects, so this is for the item recognition task, you can see how they these ones just jump around. So good, bad, up, down. See, they're erratic. This one gave up on the lexical decision test and tried in the memory test for a while, then went down and so on. And what I've taken to doing is checking my data by looking at these plots. The aging studies I talked about at the beginning, we have similar kinds of plots, and all except 2 out of 144 looked like this, like these plots, really stable data. And I think what's happening is if you've got a research assistant sitting there and watching you and guiding you, you produce high quality data. If you do it online or lock an undergraduate in a room and they're not motivated, they can screw up and produce bad data like these. I've looked at data sets in the literature, and Philip and I looked at one that was in a psych review paper, and about 25% of the data were bad, did this. And yet they were separating whether there was inhibition or not in various models. You also get experiments where there might be only 300 observations and some of them are bad like it. I mean, the field, we're getting to a point where data quality is plummeting, especially as we go to online testing. We've got to think of ways to keep it high. You can't do good modeling if your data is crap. So I did physics for a master's. And one of the things there that we learned is data and theory interact, and you've got to have high quality of both, and one informs the other informs the other back. That's why all this stuff about pre-registering is crap, because you've got to be guided by what happens. You know, you do your model, you look at your data, and then the model tells you to go and do the data or look at it in a different way. Yeah. So I've written for the first time, when you're doing a power analysis, we know that we need this many observations to get the effect, but I'm going to collect more because I want to be able to reuse these data for other modeling things. I don't want to collect the data just for the one experiment. I want to make it work. And so if I can get an hour out of a subject, I'll do that. If I need better data, I'll run them for multiple sessions. Okay, so yeah, I keep getting these signals from back there. Yeah. So let's read this very slowly then. Got 5 minutes. Discrimination in the study we did was 83% for the memory paradigm. It's about the same as the best commercial packages. So there were commercial packages from Cambridge, there were about 3 or 4 that we found. And they talk about 83% with a paired associate task. And so these results are about the same as what you can get, you know, from commercial sources. Accuracy can't be much higher because clinical diagnosis is about 90% accurate. Numeracy tasks are not as good. You've got to get control groups that look like the patient groups, except on the dimension that you're looking at. The model fit the data well, memory disorders versus controls, and we got nice effects. Online testing is hazardous, so be very careful and figure out what the data are. I recommend doing these time series plots. It's really simple. Just plot and look. I've got about 5 lines of R code that I run. So I dump the data out, run my 5 lines of code, and I can see. And then, so if you do it ritualized like that, you can look at any experiment you've run and know whether there are problems there. So can we bring modern methods into neuropsych testing? Will they help? Maybe, maybe not. The approaches to ADHD are a little bit disappointing right now. So the challenge is to use the last years of research that we've done and move it into testing. Small tweaks to existing approaches won't do. You know, it's not, you can't just modify these tests a little bit. So the hunt should be on for tasks and models that have large individual differences and specific deficits and disorders. And they have practical implications. You've got to be careful that the clinical deficits in real life may not match the cognitive concepts that have the same name. For a clinical lack of inhibition or lack of self-control may not be the same as brief inhibitory effects, say in a Splanker task. Multiple tasks that target different aspects of a deficit might be sought because combinations might produce better diagnoses. We tried that with lexical decision and recognition, and those two together didn't help, that one carried the information, memory carried the same information as lexical decision. Problem is, this research requires a significant commitment. You need control protocols with 100 or more clinically diagnosed patients and control groups if you're going to do this properly. Adding 10 minutes to a task won't be sufficient. And you need people who understand each other's research and value it. If you go and get a doctor and they say, oh yeah, I'll help you do this, that's when things have failed. Unless they're really committed, it really doesn't work and you may not want to bother. And then you need resources to provide the numbers of patients and controls to explore the tasks, measures, and models. And so we really need to do so. If we're going to be useful, I know the hub here, people here are working on practical things as well, but this is such a major thing in the US that, you know, we've just ignored it for years and years and years, and not even attack them and try and get them to change. And if you talk to practitioners, most of them, so we had a sports med doctor we were trying to do concussion with, another failure. And she had to run the impact test for concussion, but she said it was pretty much worthless as a diagnostic tool, but they had to run it because it was one of the things people expected. And there you ran on, say, visual field tests, and the data sent back, and then you get a report with pages and pages of results saying, you know, this is bigger than that, and so on. And yeah, I looked at it, and it was just taking standard tests, running maybe half a dozen trials, and then producing something that really wasn't meaningful. Yeah, and there are some references.
I mean, the word experience on top of that, it is followed up now, I guess.
Well, we want to take some questions.
Sure.
So, Roger, I'm wondering when it comes to the tasks and the way we do. So, firstly, absolutely agree on the key points here, but I'm wondering if one of the difficulties that we have in terms of isolating the individual differences is that we often do this kind of punctate testing, right? So we'll do people for an hour or something like that. So we get plenty of trials, perhaps, but that's them at that particular point in time, and maybe in a week they're going to be different, and their profile will be different. So I guess my question is, how much do you think the variability is, within the session versus across sessions?
Yeah, we've got some of those data. I think I've got 2 papers with Melvin Yap and Dave Belota on tests that are a week apart in lexical decision. We look at correlations and parameters, and we've got a lot of data that aren't published, looking at multiple sessions, and we can look at correlations. By and large, you get correlations of 0.5, 0.7 between days. So it's reasonably, it's reasonably reliable. It's not perfect. Correlation odd even is like 90-odd%. So if you break, all the tests within a session, there are differences between sessions, but they're still reliable. You still get this. So yeah, and then we also got data from some of the subjects in this memory study a couple of years later, and we were unable to find, from maybe half of them, we weren't able to predict their decline, over time, because we had the data from time 1 and time 2, and our measures just were not predictive of decline over time. So that's a weakness, and it's not clear what measures would give that. So the brain hasn't started to decline. It's not clear what the mechanism is that causes the decline, and that's just a whole nother set of questions. So that's a slightly different one to your question, but things are reasonably reliable. Why don't you just throw it?
Thanks for your results where you had 83% classification. I was wondering how that compares to status quo. So if you just do a simple reaction time test, what kind of accuracy?
I don't know. I'm not sure what. Nobody's done it like this for just simple reaction time. Then how many trials and what are your subjects? I mean, you're talking again about a multi-year study. We can get these patients. I think there are 4 neurologists doing testing, and you go in and you might get 30% of the people. We might get a couple a week. So if you're really lucky, in a year, you might have enough subjects, but then all kinds of things happen. And then the controls are important too. What we often try and do is get caregivers, and maybe 20% of caregivers, people who come in with the memory disorders and agree to do the experiments. So they're matched. But now the ones are from the senior centers and all over the place. And you just have this one screening test they have to beat to be in the control group. So number of trials, if you do simple reaction time like that, I'm not sure there are studies that have done just that. Certainly not using these methods of, logistic regression or whatever.
Why do you think it's possible to beat existing methods if no one's quantified how well they're doing?
Well, what we're doing is we have I guess these other methods, the commercial things, have like 70 to 80% accuracy. That's what they've done in large clinical trials. So we're up around that with these experiments that we ran. We're not sure you can do better, so there's an upper limit. But I'm not sure people have tried the kinds of things you're suggesting, which it might be running a simple reaction time experiment. The number line test, and we do have data now from numerosity discrimination with blue and yellow dots. And there we're still getting only about the 70% accuracy, and that's more like a reaction time task. This has a memory component, so it's higher. But in 2 other experiments we've run, we're not getting much higher in these other kinds of tasks. So it's not a complete answer, but I think there are hints that you're not going to do very much better unless you're targeting the disorder.
Anyone else? You've got the online people, Sasha?
We've got one question from Hugh. Do you have a sense of which model fitting methods, like model comparison and parameter estimation, are best for measuring individual differences? For example, avoiding assumptions about parameter distributions?
I think if you collect enough observations, they all give similar, results. We have a paper in Decision in 2015 where we looked at this in a lot of detail and compared different fitting methods for the diffusion model. What we found is HDDM was a little better than the standard programs I use and a little better than Voss and Voss's FastDM, but it very occasionally went weird. And hierarchical methods in that package assume that cross-trial variability parameters are the same across individuals. So if you generate data with differences across individuals and fit it with that, it does better for low numbers of observations, say like 100 or 200 per subject. But after 100, some of the parameters get better with standard methods, and 400 drift rates get better. So we have pictures of that. And all these methods are going to tell you the same thing at some point if you collect high quality and enough data. So I'd go for the high quality data rather than worry about collecting like 50 observations and then trying to get these fancy methods to tell you something.
I'll ask one, Roger, before we finish. Your aging data for the memory subjects shows, as I remember, narrower boundaries for the older subjects, which, is that correct?
No, it's wider for the older subjects.
Is that, I may have simply misread it in that which has the,
Could go back.
You actually had some tables where you had the boundaries.
Oh, and this is memory disorders in older subjects. Yeah, so this is not young versus old. These are the memory disorders versus the older control adults.
Okay, so they're not.
No, and young adults would be like.
Yeah, no,
0.1.
yeah, I know that there's
Yeah.
the usual younger, older. So no, it's maybe not entirely comparable. What I was actually wondering whether, and the point of the question was really whether the apparently older, sorry, the apparently narrower boundaries are actually a scaling phenomenon. It's that they have more noise. There is more diffusive noise. And as we use the diffusion coefficient as a scaling parameter, what that actually might be is an expression of greater noise.
Yeah, so you can then fix. So I've been through this. Ian Krybridge used to fit all his models with boundaries fixed and everything else. And what you found is that all the other model parameters, it fixed boundaries, noise scale, but then other parameters that you wouldn't expect to change changed as well. So fixing noise produces what I think is the best pattern of results because it's the most interpretable. Yeah, so yes,
Yeah, okay.
it's certainly possible.
Thank you. Okay, well, thank you again. Please join me again. Thank you. Roger. Ample opportunity, obviously, to talk with him and see more of Quantiles site where I